Right, so if we could get our 4 panelists up the front. So Norman, IBK, Kimberly, and we've already got Beth. So hi everyone, thanks for staying until the end. I'm Martin, I'm one of the co-organisers that's been had our job made very easy for us by the excellent events team and AV team and all the people who make all of this stuff happen. So I think it's a very interesting panel discussion, I think, here. So we want to see where we're going, what's the future, what's the big picture, what's the future of where we're going. So we're very lucky we've got an amazing panel. So we've got Kimberly Meakin, Ibuke Yoldash. Oh, how does one stop? There we go.
I'll stop now, sorry.
Apologies. Beth Simney and Norman Jepka. So I think you've probably seen a couple of them talk and maybe not all of them. So what I'll do is I'll get them to introduce themselves briefly and a little bit their journey of through being, however they started, through to being involved in bioimage analysis as you go. So if we start maybe with Kimberly.
Yep, sure. So hi, I'm Kimberly. I'm a research software engineer at UCL ARC, but I only started there like 2 weeks ago, so still figuring that out. Before that, I was a postdoc at the British Museum in their scientific research department. And before that, I worked at EMBL as a PhD student in Yannick Schwab's lab. So my route into image analysis was really doing my PhD, working with massive volume electron microscopy data and X-ray CT, doing a lot of analysis through there, finding that, I really enjoyed the software side of it. I wanted to do more of that. But I've been interested in museums, so I went and did some stuff in the museums, but now I'm working as a research software engineer, and that's my full-time job. So it's been a bit of a strange route, but yeah.
Yeah, mine is stranger, I think, but I think this room is used to that. But so hi, I'm Ibuke Oldash. I'm a member of the BioImage archive team at the EMBL EBI. And but how I started was physics and then astronomy. And astronomy, the data formats and metadata were standardized about more than 30 years ago, I would say. And as you can imagine, it makes life much, much, much easier for everyone, from the ones that produce the images to the ones that analyze or develop the tools to do the analysis and so on. And then I was involved in the in a Grand Challenge breast cancer project, which then introduced me to the bioimage analysis world and made me realize how much was lacking in terms of the data standards and the sharing. And so that's how I ended up at the BioImage archive.
I'm Beth, you met me already. I'm a molecular biologist by training, and I have no computational training whatsoever formally. I had a very fiddly project in graduate school where I was trying to measure protein intensities of various proteins at lots of little spots, and I was like, oh crap, I have to learn to code? And then I discovered Cell Profiler, and I was like, oh great, I don't have to learn to code, I have Cell Profiler. But then it turns out, then I just had a big pile of spreadsheets instead of a big pile of images, and I had to learn to code anyway. But I got really into this idea of it could be easy to take big piles of images and turn them into big piles of spreadsheets. So I started doing image analysis on all my friends' projects. And then Ann Carpenter told me that's an actual job. So I joined the Cell Profiler team as a postdoc and then stayed on as a staff scientist and now run my own group there.
So I'm a computer scientist by training and kind of by just randomly stumbled into bioimage analysis. So we started Scalable Minds 12 years ago, and we were still undergrads at university, me and my co-founders, and I met Moritz Halmstetter. He got me very excited about connectomics and told me about the large image, big data challenges that they were facing. And we saw that as an opportunity and got going. And yeah, now I'm here. Over the years, so I mean, I told you everything about web nozzles that I could fit in 5 minutes. And I think I also mentioned that we're also doing large-scale AI stuff for connectomics, where we do neuron reconstruction. So that has also been very interesting, just to work with images that are half a petabyte or even a petabyte in size, and trying to get computers to do that in a reasonable time frame.
Right, thanks to the panel for introducing themselves. So you can see we've assembled a very wide range of backgrounds and current roles. So hopefully we can get a good, thorough coverage of what's coming next. I guess we all know that bioimage analysis is a really fast-moving field. So even from the early days of BioImage and even CBIAS, the themes that people talk about have changed a lot. I was commenting to a few people earlier that the talks this year, almost everyone's mentioned deep learning. There's some very technical considerations going on. Back a few years ago, you know, every other talk, every 1 in 3 talks maybe would mention machine learning of some sort. So in really a few years, a lot has changed. It's really nice to see lots of familiar faces, but it's also even nicer to see lots of new people that we haven't seen at these things before. I think it shows the constant flux of people coming in and pushing the field forward as different requirements come along. So what I want people to keep in mind for asking these questions is there's lots of aspects to the future of bioimage analysis. It's not just what's the next deep learning model. You know, there's some interesting things, but we also want to try and dig into things like the community, technology, data availability. So we've seen some amazing examples of data sharing being a really powerful thing. Image analysis in facilities. This is something that me and Dave talk about quite a lot. So actually having core facilities for things like this is an interesting angle, and interaction with industry. So I think AI generally is this area where the boundary between academia and industry is very thin, and it's quite two-way. So we see people going both ways across that boundary. So there's a few little morsels there to sort of think about. But maybe what I'll do to get the discussion started is I'll just ask the panel if any of you have a guess for a 1 year, a 3 year, and a 10-year prediction of what we're going to be seeing coming up in the field. Who'd like to start?
I'm going to pass on 10, and that's because we were discussing at the bar last night. Image.sc is 5 years old. Cellpose is 3 years old. So many of these things that, like, Cellpose got mentioned a gajillion times the last 2 days because it's amazing, and it should be, and Image.sc has been mentioned a bunch. Those were only 5 years. This community has accelerated so fast that it's really hard to imagine. But what I certainly hope by 3 years we're a well way towards, and then at 10 years, is that we've made all of these approaches and these tools as easy as is safe to do, without then encouraging people to misuse them or use them in ways that maybe they weren't meant to be used, and therefore make conclusions that actually aren't validated.
Yeah, maybe I can add to that, which is, yeah, I completely agree. That is how I see the sort of platform that enables the wide usage of these models, but also for that, as you can imagine from my introduction, that I would mention the standardization of the data formats and metadata, because the data is only getting bigger, larger, and more basically, so we need some sort of, and I think that is where the community is going, and both in 1 year, 3 years, and 10 years, I would say, because it's not a fast or easy process, but I can see from this forum that, like, for instance, formats like OME-ZAR, OME NGFF is picking up in multiple communities, including the BioImage archive, which makes the data actually accessible, viewable, and analyzable on the fly without needing to download the huge images. So, but that is sort of both where we are going and where we actually need to go to make anything else possible, I would say.
Yeah, I guess following on with that, ideally, like, also having easier access to this really big sort of compute hardware that we're starting to need now. There's so many workflows now where we need huge numbers of CPUs and GPUs and so on. And if your institution has a massive cluster, that's great. But like not everybody does, or not everyone knows how to use it. I think increasingly, like we were seeing in Beth's talk, like these sort of web interfaces where you can have big compute architecture behind that to make those sort of algorithms more accessible for people who can't use the command line, or people at institutions where they just don't have that kind of infrastructure would also be a great thing to see in the next few years.
I don't think I have much to add to that. Of course, I'm super excited about OmiZar, and because I think for the first time we're going to have an interoperable file format that many in the community support that's actually scalable to very large image data sets. I think that's the main part that was missing in all of the previous attempts. Because I think the safe bet is, or safe prediction is, that data is just getting larger from here, and we see actually that the data sizes are increasing faster than compute hardware is keeping up. So that really forces us to develop new kind of software systems that are able to work with that. Like parallelization across multiple computers is definitely necessary in the future. It's already today, and I think people that don't have to deal with these kind of problems today will probably have to deal with that in the next few years.
Okay, so the data itself is a big concern. Do we think there's a danger that these new instruments that come along, they're just going to outstrip our ability to keep up with this, and we need to do something different? And if so, what can we do that's different than just get more data?
Yeah, I mean, I guess it's also about making sure that when we're teaching people about image analysis, we're not just teaching, oh, this is how you do it, this is how you push the buttons for the acquisition, but it's always about choosing the right tool for that specific job as well. So, making people aware of when they need to have massive amounts of data and when they don't, and like when do they need the maximum resolution or when do they not. If they want to screen hundreds of thousands of cells and they just need to be able to say, I don't know, does it express something or does it not? You can often do that with way lower resolution. You don't need that level. So I think it's partly in the training too of not always having to use the shiniest thing and the highest resolution thing. Yeah.
Mentioning training. Oh, sorry.
Yeah,
You go.
yeah, go ahead.
So mentioning training. So that's another big thing. Like, so in the audience, who has taken a training course on some sort of image analysis? So I guess a bit more than half. Yeah. So I guess this is something that also needs to be factored in, the technical training, but also the best practice and so on surrounding it. If anybody has any questions they want to ask, then feel free to just sort of jump up and wave me down, and I can either throw I'll grab this microphone to throw.
Martin just wants to throw things at people.
Feel free to jump in.
Sorry. Taking into account, that you're came from really kind of quite different backgrounds. I guess really what is interesting me for the future. So what do you think will be other area where we can overlap? So our BioImage kind of community can overlap with some other area. So I can give you some example. For example, we are working with now constantly increasing image size, right? So with constantly increasing modality, so basically dimensionality of the images. So for example, for some of my application, I found very useful go to some geospatial data analysis, right? Or satellite data analysis, because they already have quite interesting tools that we don't maybe have at the moment, or the file formats, which can contain lots of metadata, lots of other stuff. So, I think it will be very useful to, at least maybe talk to those people. Do you have any other examples of other, areas, and why they will be what can be the connection points?
I just wanted to say that in the ZAR community, also the geospatial people are very active, and I really like that about ZAR as well. That community is using that. Bioimaging community is hopefully taking it up even more now, so we can really work together and figure out metadata and figure out common problems and solve them. Yeah, so that's great.
Yeah, I perhaps have an example in the other direction. I was working at the British Museum before, and just also that other fields can benefit bioimage analysis, but also bioimage analysis can benefit other fields too. So with museums and cultural heritage, they do loads of microscopy and they really need this kind of image analysis input, and loads of the tools that we use are also really useful for those kind of problems. So I think, yeah, you know, when you have a microscopy conference, it's worth inviting people from more out there fields that you might not imagine as well. But
I guess astronomy is one of the obvious answers. Like, I mean, that's how I ended up with that Grand Challenge project, for instance, and the images actually look a lot alike. And there was a Murphish talk, well, not a Murphish talk, but a talk mentioned Murphish, where the background fluorescence, for instance, is something that you can see in the images of nebula, for instance. So even before the whole, you know, this technology was there, we were seeing same kind of images in the sky. And the astronomy community is also, I think, looking into XAR because of the, you know, ever getting bigger size of the images, I would say.
You can expand the spikes also for the stars, not only for a single cell.
Yes, yes.
Maybe if we go to the back microphone for a question. Oh, yeah.
Thank you all for the great talks. I'm a biologist, but I'm really enjoying the talk so far. I have a question as someone who follows from afar the evolution of bioimage analysis, and I'm thinking of my colleagues that maybe haven't really started doing image analysis, but they want to start. And what I feel is overwhelming for them is the abundance of resources, and there is no proper, at least, guide or some sort of index where they could find all of the crazy softwares, all of the crazy resources. So I'm thinking for the future, will we be at least creating or finding like a website where we could like have at least links to all of these different resources? I know that there were some effort made with the BioImage informatic index, for instance, where you can find like different things. If I was like just looking for, I don't know anything about image analysis, or I know a bit and I want to analyze nuclei, I would want to be able to go to an index and say analyzing nuclei and all of the solutions that are proposed. Will that be something that the community is working towards for the future?
I mean, BIII is definitely the closest thing we have to that. It turns out that just a giant list of things in terms of approachability, I mean, you need that, but it turns out it's not the most approachable. It's a hard problem because there's so many different things you might want to pull out of an image, but I know one thing that I'm excited about is the ability to sort of, as we're demanding people attach metadata to images, is the idea of, can we just ask people what they're analyzing and then say, all right, nuclei, so those are things, nuclei at 20x from human cells are about, 80 pixels across circles. So let's go into our database of all the 80 pixels across circle things. And so we actually proposed an initial metadata spec for that in terms of problem categories. And I think things like that, in addition to just sort of cataloging what's in the image, cataloging what do you want to know, are going to be really helpful for doing that sort of recommendation.
I was just going to get in there as well. And just, I think, yeah, it's really difficult to have like a one sort of flow chart of like, if you have this, do this, then do this. And I guess, like we've all said already, a good starting point for this sort of thing is the forum. If you can post what your question is, you're somebody who's never done it before, you can give some small examples. Most of us are on there and would be happy to hop in there and give suggestions. So that's perhaps an easier way to get some custom examples if the big list doesn't help you at first. Yeah.
I think having multiple avenues in as well, so there's no single thing that's going to work for everyone. So as long as they're somewhat joined up and things can go between them, then that's maybe a necessary first step before we go for the all-in-one.
Yeah. Martin knows this because he's also on that paper. One of the things we found in the survey was, different groups of people want very different things. Developers hate videos. Beginners love videos. And so developers don't make videos because we hate them, but beginners want them. And so putting out information in written and video and, this like all different kinds of ways is the only way we're going to get to everybody.
So I guess the title of the session also make reference to the last summer issue of NATO Method, talking about future of bioimage with fancy microscope, where you could talk to your microscope acquiring data for analysis, all that. But I have most modest wishes that would be standardizing image formats that I could trash bio format. I don't need it anymore. Also that I could maybe have standardized way of inputting data to my neural network, so I don't spend hours converting inputs, outputs just to make it a lot of useless parts of the work. But so here's my question: that which one do you think will arrive first, then the fancy microscopes or the standardized way, and why is it the fancy microscopes?
I would say, actually, if we start sharing the data more, then the fancy microscope can slow down a bit, because at the moment what's happening is that if you don't share your data, right, so everyone wants to repeat your exciting results, or like, oh, you know, I can do something with this kind of experiment. But as Beth was saying in her talk, there is a lot of things you can do with one image or one, you know, set of images that the original producer haven't actually done. So if you make your data public via public archives like the BioImage archive, then there is a much more chance of having it reused, and therefore like putting less sort of time and energy into having fancy microscopes and experiments. And also this is a way to sort of standardize the metadata and data formats. So for instance, we implemented the RAMBI, the recommended metadata for biological imaging guidelines, into our submissions. And so, you know, we are working towards the same for the annotations, like segmentation, masks, class labels, and so on. And we are like working towards, I mean, there is the poster. Unfortunately, this is the last session, but if you go to the BioImage archive website, you will find the new feature of galleries, where you will see the OMISAR images, and there is one specifically for AI, where we are curating a little bit for AI-ready, our AI-ready data sets, so, and presenting it in such a way that it is much more standard and accessible. So I think these are, like, for instance, one of the things that would kind of help guide the community into the right direction of standardizing both the data formats and for input and into and output, so to say.
There's just a lot of moving parts in building standards and getting communities to agree on things and building consensus. And once they agree, then you still have to implement everything. So it's just a long process. I think I'm quite optimistic with OMISAR that we've now taken a couple of big major steps. But yeah, it just takes a long time. And I mean, I don't know how many people in the room here are already using OMISAR for some of their work. Yeah, okay. I hope many more of you know about it. But actually making the jump to using it, even when the standard is kind of ready, is a big jump for many people. And because all of the parts need to be in place to actually make things better, right? Because otherwise you just have one more file format that you have to deal with, and nobody wants to do that. So, yeah.
I think Stefania's got some. Oh, sorry,
No, I was just going to say, I think also some of the emphasis on hardware versus software has been a bit about how funding schemes used to work. Like, it was a lot easier to say, I want to buy this microscope, than to do something looser about, I need money for designing community standards, and we need to do a meeting, and then we need to do this. And yeah, that's also part of it. It's been changing recently, which is great. There's been a lot more funding available for that sort of thing. Could still be more, but, but yeah.
So I think Stefania's got some questions.
Yeah, I have a couple of questions from online, and I think one really segue nicely with what Kim has just said, relating to web interfaces, basically allowing for democratization of technology, very hard word, sorry. And how can we afford this? So would institutions, government, and organization have to build this infrastructure together? And another aspect would be from another question, but I'm putting them together. How is the landscape changing when like a big company such as CZI, that's the example they bring, decide to start funding and invest in research?
So the web tool thing, web tools cover the whole spectrum. So some of them are, there is a big giant server that sits somewhere and you have to upload your data. And then, yeah, some of it, like what I just showed, all of the web tools I showed, they're running on the user's local equipment, which has its own like advantages and disadvantages if your local equipment sucks. But absolutely, we need to start showing the funding agencies and stuff, and I know there's movement around this to sort of say, like, we need more investment in Galaxy and imaging tools for Galaxy and things like that, where we can make it easy. You know, Band was a great example that Tishy showed yesterday. And you called out CZI specifically. I will give them a ton of credit for sort of coming out and saying community building is important, maintaining existing software and not just writing the 27th copy of Fiji, is important. But CZI is trying to sort of plug a hole in a bucket that is a very big hole in a very leaky bucket. And so there's a lot of work by a lot of smart, dedicated people to try and get other funding agencies to realize this is a problem. But so far, it's not great.
Yeah, I would say there has been some discussion among at least a few of us about providing this image analysis infrastructure as a service, like the databases or repositories. So I think, yeah, that's definitely a good point, especially again, like, not everyone can afford that kind of stuff. And yeah, I think, as Beth mentioned, there are some developments towards that direction, but yeah.
I just wanted to say the web is great, and because it makes it easier for people just to try things out and set up, like all of this Conda stuff, or maybe you don't have to deal with necessarily on the web. And also it's just a technical enabler for scalability, because in the end, from a user perspective, it doesn't really matter if it's running locally on your computer or if there's like a massive cluster or the cloud that sits behind that, and users can tap into that compute power without necessarily knowing anything about the infrastructure. I think that's super powerful, and I think that's underutilized in bioimage analysis so far.
Just comment also on that about the Galaxy stuff. So I mentioned yesterday there is this NFDI for bioimage. So in Germany there is actually funding of a consortium, and actually quite a bit of is about making image analysis in Galaxy a reality. So let's see. And but they really have a lot of stuff. I mean, they have all this solved, that you have the compute infrastructure somewhere and so on. I think what they are still, like, getting the data to the compute is still always a bit tricky, but I think they have even fancy stuff, like you can register your local HPC at some Galaxy thing in Sweden, and then it will cleverly know that it runs on your computer, because this is, I mean, so they have really, so I'm just saying, before you do your own stuff, maybe check Galaxy. So I think they have quite some cool things, but it's still very early. I think right now you can't do even a Gaussian blur, as far as I know, but or maybe you can, but like just providing stuff in the web, I think it's a pretty mature platform. So yeah.
Okay.
So talking about the 10 years horizon, do you think you will still be in the job market? Or because of this, a large language model, will take over and they will be able to do your job? And what could be the kind of input you can give? Thank you.
We've got quite a long way in before large language models came up. So who had that on the sweepstake? I don't know. Sorry, go on.
Yeah, fortunately, there are enough problems to solve, and I don't think a large language model will be able to help with all of them. That being said, I think they're super interesting, especially to making it easier to write scripts or stuff like that. I think that's low-hanging fruit. But we'll also discover more things that these kind of models can help with. But I think the more interesting question is actually where the large image models going to be? When do they pop up? So that, I don't know, you have this one big model that can solve a bunch of different tasks. big ones. I don't know. I don't see them on the horizon anywhere, but maybe on a 10-year scale, something pop up.
The first CZI imaging meeting, Alan Goodman, who's the developer of Cell Profiler, stood up and said, my 5-year goal is for some people to use Cell Profiler less. Because and then went on to explain it's because classical segmentation. He's like, I don't want to help people do that anymore. I want there to just be models that will do it. So I certainly hope AI comes for parts of my job. I mean, I love working with users and teaching them how to do it, but there are things that are too hard that could be easier. But I think the second we come up with an image analysis way to solve X, clever wet lab developers are going to come up with Y, and then we're going to have to, figure out how to solve Y. So I don't think we're running out of, I think we're going to just keep making more problems to solve faster than we're going to solve all of them. But
I was going to say for the large language models, for using them for coding, I think like anything else, they're going to be a tool that gets used. I don't think it's going to replace, all of the image analysts. Perhaps it helps to remove writing a lot of that sort of boilerplate stuff, or getting into a new language, you might be able to get there faster and so on. Like, I think there's a lot of good stuff out there. I mean, I think the other issue of them right now, especially the really big models or the image ones, that there's still a bit of an open question about where the ground truth and the training data comes from, and whether they should be able to just use anything off the internet without permission licensing and so on. So I kind of feel like it's still in a bit of a gray zone for those really big ones, but there are more and more that are trained on specific data sets that have great uses. So yeah, I think it's a mixed bag, but I think it's always going to be a tool that sort of enhances alongside what you already have rather than replacing it fully.
Thank you.
And
I think
Stefania?
there's one more online relating to OMISAR and standardized format, asking, can we as a community tell microscope companies that we want to stick to a new standardized format? Or basically, I'm rephrasing, but we'll always have to go through conversions.
Yes.
Yeah,
Please.
yes is all
Yeah.
common answer, I think.
As
Unanimous.
often as you can.
there was a interesting article that Jason Swedlow and Josh Moore had both some very funny comments on in Nature recently talking about that. But the fact of the matter is we have to convince companies that it's worth it. They've all spent a bunch of time developing these formats, which presumably they think have value. So they at least need to have a seat at the table. But unless we we can ask and we should make it as easy for them to do it. But unless you say, I'm sorry, I'm not going to buy your light sheet if it doesn't write czar, they currently don't have a ton of incentive to do that because they know you're going to buy the light sheet anyway.
And the formats have to support all of the requirements that the microscopy companies have, and that's why it's so important to get them at the table. And I think they're very much invited.
Yeah, and I just want to add, actually, there is also just want to give a shout out to the Quaret Limi group, which is trying to do the same for the metadata. And they are actually talking with the microscope companies to have the imaging related, microscopy related metadata put into the image at the microscope site. So as long as there is, community effort, I think things can go in the right direction.
So what are the actions that we as a community can do to make this happen as fast as possible? Do we just go and knock on their doors and say, give me only TIFFs, or do we have separate meetings, or how do we engage with industry on this in a way that benefits everyone?
Yeah, I guess so. Like as I mentioned, Quaret Limi, there are some existing groups that are doing that. So you can start with joining those groups, but also, as Norman was saying, developing, and Beth as well, developing the tools that would enable the transition, or like working with the microscope companies, the tool developers, I would say, and the microscope companies working together to make the read-write bit, especially for the data format standardization, easier, is, I guess, like one of the necessary steps.
And I guess we could target a bit, we have an example here, is the smaller, more agile companies that are able to do this sort of thing to demonstrate that this is a good way of doing it. Maybe we can follow Norman's example and
Please do, yeah.
Yeah, and the imaging facilities, I would say, because they are big providers and also big customers.
Yeah, I mean, but as long as something like bioformats exists, and as long as we keep maintaining things like bioformats, again, if you have the choice between spending, putting two software engineers for a year on making your, proprietary file into a czar, or you could not do that, So we as a community are going to have to decide it's important enough to us that we want to not buy the light sheet or, figure out or stop making bioformats or stop doing
Yeah.
something, because we have to make it worth they have to be able to go to their bosses and say, I'm going to put two software engineers on this for a year to do this. And this is why, for a business perspective, this company will make more money if you do it.
Yep.
Making a step back to the previous question about language models, do you think we are close to the point when I don't need to do the work twice? For example, annotating data and then writing the method section, or writing a script and then writing the method section? Essentially, like when Cell Profiler can write a method section instead of me from script.
Has anybody tried that? I mean,
There's a Fiji plugin, right, that does something like this.
Yeah, there is a. Mhm. So part of the problem is just also it's a higher level of understanding. Cell Profiler will spit out a list for you of all of the modules that are in there, but what does each one contribute to a larger sort of philosophical goal? There's a million different reasons why you might do a Gaussian blur. How is Cell Profiler going to know that you're doing a Gaussian blur for reason X as opposed to reason Y? And so I think making tools work more in that way, where we don't require users to know about atomic operations, but we make it more purpose-driven and question-driven, is I think the only way we're going to get there. Because if it keeps being atomic and we keep saying do a Gaussian blur and then a threshold, it'll never know why.
I was just wondering where you think the sort of spatial transcriptomic technologies that rely on imaging, where do you think those fit in? Are those in the sort of the bioimaging world, and the fishmerscope and all the other technologies that are all evolving?
Yeah, that's a very good question. And I don't have a clear answer, but there is a community and institutes wide efforts trying to come together to make more sort of standardization and one point entry for spatial transcriptomics data sets, at least in terms of the archives. And I think in terms of the data formats, so our collaboration with the Web Atlas team, as you've heard from Tong's talk, I think there are ZAR formats for transcriptomics data as well, the NData ZAR and so on, which is going towards the a merged direction, I would say. And that is one of the next big things, right? So one of the obvious multimodal data world. Yeah, there are efforts, but I don't at the moment have any clear answers.
On to the next one.
Again. So that's going to be again on metadata. And so when I went to the conference, the ELMI conference this year on imaging world, it's a lot of topics on data management and how we're going to organize our data, annotate them better. And that's work to do at the moment of acquisition. But I feel that's also a topic less taken by the bioimage analysis community overall, that it doesn't matter as much that the data is organized, annotated. But so what do you feel the role should be of this community to help also push toward this data management practice, and then push on all these standards and help overall? So also linked to the other question earlier.
I think if the metadata were all in place, it would make the life much easier, because then we would have to ask less questions, right? Because if like the metadata already contains what experiments are about or how they've been taken, then that could facilitate just the image analysis process, I would say. So BioImage analysis could demand metadata basically that is in place, and I think Tishi did that at least with the file name schema. So that's maybe a good example. I don't know, or a starting point. Yeah, and that being said, also during bioimage analysis, lots of metadata is being created, and there's no clear way of how we should store that. And I know the annotation team at BioImage Archive is also looking at that. So I think that's also underexplored and just something that the community needs to work on.
Yeah, I think that's one of the missing things from the OMISR at the moment, for instance, that there isn't the a standard way to put metadata into the OMISR headers. And like, I think the community, including us, we are working towards something like that, a framework, and like to have to implement REMBI into the OMISR headers, and then like putting all the down the line analysis metadata as well. So if you so this is one of the things, nice things about the astronomy data format, right? We have everything that is related to the data is packed together with, so the metadata is packed together with the data, with the image or the tables. And I would say this is where we should head towards, because image, I mean, as nice as it is, file names are not metadata, because anyone can change them, and you won't have any track of, you cannot keep track of that. But like, if you have all that information somewhere packed together with the images or packed together with whatever data you have, then,
So what I mean also by data management is a way to share your data, collaborate, and organize your experiments and your data, binding them together. And these are different aspects than also metadata tracking.
Mm-hmm. Yeah, I mean, so I think part of it is the professional bioimage analysts a lot of times are not the ones making or long-term storing the data. If you're in that in-between role where someone's like, I made my data, it's my long-term responsibility for it, the analyst isn't involved in that ultimate decision-making of where it gets archived. All that being said, there's definitely a lot of analysts who are thinking about this a lot. There's going to be a thing around data management and organization and stuff next week, and I'm on the metadata and data management subgroup, and 3 of the 4 moderators are image analysis people. So professional image analysts as a whole may not be thinking about that much, just because, again, we aren't ultimately the keepers of the data, but certainly people are working towards that.
I was just going to say, like, from the training deep learning model perspective, I think we've all had this experience of like, you're looking for training data and you download some from here and some from here, and every single person has a different format and it's in a different folder structure, and you spend half your time trying to get everything in the same in the same format. So yeah, having some sort of standards for how to arrange things would also make that a lot easier, so that you can download from multiple places and easily access it and start training things right away.
Because it's in the interest of the software developer to not stop caring about how do I write my data loader and how do I save the data, but it's things that exist in data management already.
Yeah, and also the data management systems, they will also benefit from an interoperable file format, because then you don't have to, first of all, support all of these file formats, but also it then makes it easier to basically keep the data inside the data management software, and then just for some kind of analysis task that you want to do, you basically just stream the data out into that tool, and then the results can go back into the data management system. So yes, it can be much more integrated, and that makes it also easier to track metadata and keep everybody in the loop.
Thanks.
So Dave's just given me the nod that we can have one more question, and then we'll have to wrap up. So go for it, nice and quickly.
Yeah, thanks for the nice discussion. Yeah, I just since the question came up, so I'm a software developer working at a microscope manufacturer, Luxendo, and yeah, I mean we are very interested, in the OMISR, and we've been also following it a long time, also taking part in some discussions with Josh and so on online over the last years. And we've also invested a lot of time, like developing consistent metadata, also that's consistent among, for example, raw images, but also already fused images, right, to make it all compatible and enable already processed images being fed back into the processing pipelines and so on. So, yeah, I think we just should keep working together because it would also solve a lot of our problems, right? We're also having a lot of problems with HDF5, for example, being like a bottleneck. And so, yeah, I think as a community effort, maybe can make it work. Thanks.
That sounds great. Yeah.
I think that's a great point to finish on, so thanks very much. Thanks to our wonderful panel, Kimberly, Ibuke, Beth and Norman. Thanks everyone for all the stimulating discussions, and we'll thank the panel again, and I'll hand over to Dave.